So without any delay, I'm happy to introduce Joel Dudley again, Director of Biomedical Informatics, who will introduce our next keynote speaker. Joel?
All right, thanks for coming back, and everybody. We got a really exciting keynote here. I'm happy to introduce Atul Butte. There's many possible titles you could attribute to Atul. He's a pediatric endocrinologist. He's an expert in bioinformatics. He's a multi-entrepreneur scientist, so he's taken, which I won't steal his thunder on that, but he's taken NIH funding all the way to startup to venture capital funded. So he's done it all, done all the things I think a lot of people here are aspiring to do, and it's just getting warmed up. So I won't embarrass him too much, but he was just elected recently, within the last month, to the National Academy of Medicine. So it's pretty awesome to have informatics people starting to get it into the National Academy of Medicine. He was awarded the Champion of Change for being an open data pioneer directly from President Obama. He's given talks at TEDMed and things like that. So you might have seen his face before, maybe, if you pay attention to that. He's currently director of the Institute for Computational Health Sciences at UCSF and director of clinical informatics for the whole UC health system. And he overcame one of his greatest challenges in his life, which was having me in his lab for 4 years as a PhD student. So he's still here to talk about it. So thank you.
Excellent. Well, thanks for having me here. It's great to finally be over here at Mount Sinai. I've seen Joel's lab before, but never on this side. Well, thanks for having me, and thanks for sticking it out all the way to the end. So first, I'm a medical doctor talking to a pretty medical audience, so I got to start with my conflicts of interest. Just a few. So suffice it to say, I've started a bunch of companies. I consult for a bunch of companies. You probably don't want to believe another word I say over the next half an hour. Wouldn't blame you. But I'm most proud of the bottom right. These are all the companies started by my students. More than half my graduate students now start companies, even if they go into academia. And they do this with the most amazing platform in the world. And that's data. And it's often big data. It's often big open data. So I'm going to show you how they do it. I'm going to show you how I do it. And maybe I'll convince you that this is the most amazing time to be in biomedical research if you actually start to think about using that data. So a lot of people use a slide like this as a slide of shame. And I use this as my slide of significance. Because I've learned now in academia, if you want to change the world, you can't just keep writing papers about it. You have to actually do something and take that technology out of your institution. So I'm going to talk a lot about this kind of thing. It's not taboo for me to talk about it, okay? I actually take pride in this. So if you haven't heard by now, we're in the data deluge. My favorite example of this is the cover of The Economist magazine from 2.5 years ago, this guy collecting ones and zeros with the umbrella. And as of 2.5 years ago, they pronounced that the human species is now generating 2 zettabytes of data every year. If you forget your metric prefixes, it goes kilobyte, mega, gigabyte, terabyte, petabyte, exabyte, zettabyte. And then, of course, the following year, it'll be 4 zettabytes of data, because, of course, this is doubling every year or two. And I'll be the first to admit most of these zettabytes of data are these videos of kittens playing on pianos and yarn, right? Entertainment value, perhaps, not scientific value, but there's scientific data in the zettabytes. So a Large Hadron Collider generated petabytes of data to come up with data for new subatomic particles. But my favorite example is NASA. So NASA announced about a year ago that they will have so many telescopes pointing skyward by the end of this decade that they will be generating an exabyte of data every day. Every day, so you can see where exabytes are on the scale. Every day. So what's so interesting about NASA's exabytes? They already generate so much data, pictures of the sky, that there are not enough astronomers on the planet to look at all these pictures. So what do? When you're overwhelmed with data, you get your friends on the internet to help you. That's crowdsourcing, and you can sign up at galaxyzoo.org, and you can take a test, and then you can say, is this a star, is this a galaxy? But something telling, when you run out of scientists, you need to involve your friends on the internet, right? Crowdsourcing, citizen science. And of course, I'm going to tell you, I believe we're already there in health and life science. And why are we there in health and life sciences? Because of gadgets like this is just one representative of many high-throughput technologies that are out there in health and life science. This one happens to show the best, so that's why I love Affymetrix. I got my start about 15 years ago in this field with some of the first of these arrays on cancer when I was up in Boston. Now, these chips are amazing. In fact, I carry one. They're so cheap now that I carry one in my bag. So this is what they actually look like. I have one in my hand here, right? This is actually one of these chips. They're so cheap now that you can put a sample of cancer, diabetes, normal tissue, animal models. You get a readout of the entire genome, right? All those recipes, how much of this one's getting cooked, how much of that one's getting cooked, all the RNA. And then when you get tired of measuring them one by one, you measure them in the 96-well format. And that's cheap enough now that I can carry that in my briefcase. And then when you get tired of measuring them in a 96-well format, you measure them in the 384-well format. And that I have in my hand. That's cheap enough now, right? Visually, this is exponential growth in data in life science. I cannot explain it any better way, right? We move from 1. We don't scale like 1024 like life science, right? We scale with this weird multiple 96, right? 96-well, 384-well, right? This is exponential growth in life science data. Now, I got my start with just the first 60 of these arrays. They were priceless at the time. Now these chips are like $200 or so, but that's not the most amazing part of this chip. The most amazing thing happened because everyone started running these chips. And I'm just using chips. I mean RNA-seq, and I also mean DNA sequencing and mass spec and proteomics, all those technologies. So many scientists were using this technology, everyone started writing papers about it. Here's a list of genes changing in that cancer. Here's a list of genes changing in heart failure. The journals started to get overwhelmed with these papers, and the journals said, hold on a second. No more papers with these chips. No more. Enough with these lists. Unless you put this data into an international repository so the reviewers can double-check the math. Getting important to double-check that math as we get closer to the clinic. And maybe others can use these samples for something else, right? The precedent for this goes back to GenBank, which is, I think, 40 years old, right? No one wants to type in a DNA sequence from figure 1, right? You got to electronically share this kind of stuff. If enough scientists use the technology, you got to share that data. And fast forward, right? Fast forward to August of 2012. There's a special article in Nature, interviewed my lab and others. In August of 2012, we hit 1 million of these chips publicly available now. 1 million of them. You can see the growth trajectory from 0 to 1 million. But if you look really carefully, it kind of looks like it's slowing down there, right? You see the slope there? And then you realize they only counted half the year that year, right? August 2012. Still doubling every 2 to 3 years. If you go to these repositories today, 1.8 million samples already run, already digitized, waiting for you. Because no one else is there to get all this data and do something with it. What do most scientists do? They get another grant to go collect more data, right? The data is just sitting there waiting for you. Now, you can get lost in the millions and billions and trillions, so let me make it really concrete. Any one of our scientists, actually any one of your scientists, can go to these repositories. Maybe you're studying breast cancer, colon cancer. You can just search for, forget about our scientists. Any high school kid now that needs to do a science fair project, she can go to these websites, right? She needs to do a science fair project, let's say on breast cancer. She can go to the website, search for breast cancer, and find and download 60,000 samples of breast cancer about as easily as she can find a song on iTunes today. What an amazing time it is to be in biomedical research. 60,000 samples. If you haven't figured out why that's a magical number, right? That's more samples of breast cancer that high school kid has than any breast cancer researcher, because every one of them has to deposit their data into the middle. And whoever figures out the middle has more data now than any one of the contributors. And if it's not breast cancer, it's colon cancer, it's prostate cancer, it's diabetes, whatever disease you want, probably some scientists already studied it for 15 years. The data is just sitting there. Actually, above the 59,000 number, there's a number 2,400. That's the number I like better. 2,400 independent labs have already contributed experimental data into the system. Let me tell you what a ridiculous number that is. Let's pretend I'm a brand new researcher in breast cancer. I'm a newbie, okay? I'm a newbie. And you're all the people reviewing my grant, my NIH grant. And let's say I propose in my grant to you that I'm going to get 2,400 of the best labs in the world studying breast cancer to get their best characterized patients to run their latest technologies, and I propose to you they're all going to give me all that data for free, okay? You would laugh me out of the room if I proposed this to you, right, in a forward direction. Yet here it is, right? So when I think of public big data like this, I call it retroactive crowdsourcing, okay? What do I mean by that? 2,400 labs are there to help you, and they don't even know they're helping you. Actually, 2,400 labs are there to help you, and you don't even know they're helping you. And for some reason, I don't know, 60,000 samples are enough for you, they're not enough. Just wait another year or two, it'll be 100,000 samples. In this half an hour, we're gonna add another 100 samples, right? What an amazing time. We are living in this amazing time to do biomedical research. Yeah, I know NIH funding is dropping and all of this, and then they give you 1 million samples to get started, just waiting for us, waiting for you. Now, if you have any doubts a high school kid can do it, here's the high school kid that did it. I've yet to meet her. I talk about her in all my talks. This is Brittany Wenger from Sarasota, Florida. She's now a Duke undergrad. And at 17 years of age, she won the top prize in the Google Science Fair competition. She's carrying the white trophy there because she's a 17-year-old that built an artificial brain, neural networks, machine learning, to diagnose breast cancer. She had off the internet data, she built a classifier, she won top prize. I guess she was bored, so the following year she did the same thing for leukemia. And she was in the paper again. Of course, what I mean is if a high school kid can do this, all of us should be able to do this at this point. Certainly our researchers in our institutions should be thinking this way. We're leaving samples on the table otherwise. We're leaving money on the table otherwise. We give away this data. But somehow we think if it's free and on the internet, it must be valueless, right? Like all the other kitten videos. This is the most valuable data in the world that we put out for free. So what do you do with all this data, right? So our first big story was, can we use some of this data to get to drugs? And you've heard a lot of different speakers and a lot of different stories of where things and innovation is going to happen in drug discovery. And then I see graphics like this. So this article comes out every year by Matthew Harper in Forbes. And this is an article really about how much it costs to come up with new drugs. If you actually ask lay Americans, they actually know this number. And that's funny. Lay Americans don't really know much in common if you think about America. But they know it's $1 billion in 10 years to develop a drug, right? Because they advertise this on the Super Bowl, all right? So that's why most Americans know this number. But it's an underestimate, okay? If you look at that third column there, how do you really get the real number? Pharma R&D, divide by the number of drugs. It's very simple math. For these top 12, it costs between $4 billion and $12 billion to make a new drug. Kind of not sustainable. Kind of not sustainable. And so when you realize, look, I showed you on my first slide, I kind of consult for them all, okay? I love them all, but I'm losing faith that they're going to solve this problem. And then you realize, well, as the medical doctor in me, where are we going to get our new drugs from, right? Where is all this innovation going to happen? And you realize it's going to have to come down to us to do this. It's going to be a little guy, little gal in academia, in the startup companies to figure out the new way to do this. And can we do this with public data? So that's really the whole story here. My first big NIH grant was, instead of cancer or diabetes, why don't we just go get every human disease studied by these chips? Just go get them all. And on the left there, it's an example. We get disease samples and healthy samples, disease and healthy. On the right, we started to see people depositing data on drug experiments. Here's cells with the drug, without a drug, with the drug, without a drug. And we put these two together. The reporters for this called this Match.com for drugs, okay? And what's a famous saying when you're trying to find a spouse or a mate, right? Opposites attract. So I'll show you all the statistics with my arms. The colors there represent genes and the recipes, how much they're making, but let me just use my arms. If I've got a disease where this gene goes up and this gene goes down, and I can find a drug that can make this one go down and this one go up, maybe there's a match there, right? I can just find a drug that naively reverses what I'm seeing. This is 2 genes, imagine 20,000 genes. And we turn our crank. And we got lots of ideas. And we got lots of ideas. This chemical might work in that disease, that chemical might work, but where we got a lot of fun ideas, it's not just a new drug, but how about a new use for an old drug, right? A field called drug repositioning. We didn't invent this field, right? But boy, can you use the data to do this? Instead of the famous examples of great side effects which became drugs like Viagra and minoxidil, right? Can we actually come up with drugs on purpose that actually seem to have another effect? So what do you do next? Well, you got to run a clinical trial, right? And all my friends scatter, right? Because they know I'm going to be proposing a lot of trials. But actually, before you put a drug into a patient, you got to actually test it in animal models. Whether you like them or not, we got to do this, right? Animal models. And all those friends start to scatter. A few of them really help us a lot with some animal models. But what do you do when you get stuck, right? The computer comes up with so many hypotheses. How do you get these things tested? I have a lot of friends. I have a lot of collaborators. But when I really, really need an animal model tested now, I go to something new, something called assaydepot.com. Kind of sounds like Home Depot, and you're right, okay? What do you want to buy today? Biology, chemistry. DMPK stands for Drug Metabolism Pharmacokinetics. If you don't know what that stands for, don't click on that one. Pharmacology and toxicology. Well, I don't know, I need a mouse done. Let's click on pharmacology. What type of mouse do you want to do today? Bone, cardiovascular, dermatology, diabetes, general, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. I'm a diabetes guy. Let's click on diabetes. Okay, here I can order a 16 mouse experiment. OB, what is that? If you read there, it says this mouse has been eating a lot and getting diabetes since 1951. This is a standard model for American diabetes. You eat a lot, you get diabetes, right? And in 4 weeks, 28 days, this company will do the fasting blood sugar, insulin tolerance test, glucose tolerance test, and all the results come back 28 days later, right? What am I covering up the price? $9,000 for this service, 9-week turnaround time, and literally, add to shopping cart. This isn't the hard part anymore. Now, there are a couple of you in this audience I know that do mouse models. You know some parts of it can be more sophisticated. And then there's this, where these kind of common translational models, you can just buy them off the shelf with a credit card off the internet from a contract research organization. Now, some of you with really sharp eyes see this as being done in China. Some of you have a problem with this, others don't. I don't. But actually, when you hit add to shopping cart, you're shown all 133 core facilities or companies that do this experiment for you, right? Maybe you need the FDA-approved ones. Click on the bottom there, 5. ISO 9001, there's 7 of them. Triple ALAC, 28. You see some are in Maryland, some are in Wisconsin. Outsourcing doesn't always mean offshoring, right? You get to pick and choose. But for dry bench scientists like me, this has been incredible. I can work in diabetes 1 week and cancer the next week. I don't have to love these mice. Of course, we've got to design all the protocols, but they do all the hard part. Now, some of you are wondering, how do you actually trust these companies to do anything right for you, right? All that computer stuff is one thing. This seems like the most important part, because if this works, you're going to put in humans next. And I will simply say, if I really, really need to trust these companies, I just simply order 2 of them from 2 companies. Hell, at $9,000 each, I can order 10 of them from 10 companies, right? How many mouse researchers do you know when they get their experiment to work? Do they go across the hall to get a completely independent team of researchers to reproduce their findings? You know they don't do that. But what I've just subtly done there is I've converted this quality problem that's in your minds, and I now make it a function of dollars. You have more dollars, you get better quality, right? I don't trust any one of those experiments on the internet, but I trust what I see in common. I don't trust any one of these companies, but I trust what I see in common. I can get more than just one of these to work. It's wisdom of the crowd, but in a data sense here. And this is literally what we do. We might order one from the West Coast, one from the East Coast. We don't discriminate against Chicago, but the flyover states, but we tend to do East Coast and West Coast. And the famous example that we have is this seizure drug that we figured out. Topiramate works on Crohn's disease, inflammatory bowel disease. Of course, Joel Dudley was involved in this. And these are our famous rat colonoscopies. Not enough of you are chuckling, though some of you are over age 50. You've had a colonoscopy, so let's illustrate the geometry involved. This is a colonoscope. This is a rat. This is what you don't want to learn how to do in your lab. You find a company that knows how to do this, right? And here are my rat colonoscopies, right? The left rat is a normal, healthy colon. The one in the middle gets a chemical called TNBS to give it inflammatory bowel disease. And on the right, it's all better with the seizure drug, 40 megs of video with this Science Translational Medicine paper. So I'm not just outsourcing to save money. I'm outsourcing actually to get the best damn data in the world. There's a company that knows how to do this. We had no collaborators on our campus that knew how to do a rat colonoscopy. We've done more than a dozen of these. This is my favorite one. This one came out last year. A psychiatric drug, imipramine, seemed to work against small cell lung cancer, right? Now, small cell lung cancer, humans smoke to get small cell lung cancer, 5% survival rate. This mouse, you knock out these 3 genes, and it gets its own lung cancer. This is not a xenograft. You can see the nodules in the top left, the little cancer nodules. And on the right are the panels of the same kind of mouse treated with imipramine. Imipramine is an antidepressant. We still have it in the pharmacy. It's a tricyclic antidepressant. We don't use it anymore because we have serotonin reuptake inhibitors like Prozac and others. And we don't use imipramine anymore because it's got some side effects. In some people, it makes you really sleepy. And in others, it might set you up for QT prolongation, might set you up for an arrhythmia. Actually, neither of those 2 side effects sound as bad as having lung cancer. 5% survival rate. And the cancer is melted away. It's gone in this mouse. Now, why do I love this one out of the dozen or so we've done at this point, right? We start with public data, right? We get this idea this drug could work. We test cell lines in a petri dish. I'm not even going to show you that. We got this amazing picture of these mouse lungs 15 months after the prediction. We get IRB approval. We launch a clinical trial. Patients are dosed on this. Total cost of the trial, $50,000, not $1 million, not even $100K, right? This is one of the seed grants from our CTSA. I don't know if the drug is going to work or not. It's actually probably not going to work because of those 2 side effects. I'm learning. The sleepiness is the worst of the 2 because if you've been told you only have 5 months to live, you don't want to spend it sleeping, okay? It turns out, we're chuckling about that, but it's a big deal, right? It doesn't matter if this fails. We got to run more of these, and we got to do it cheaper. And they're not all going to be repositioning, right? But we got to get to faster stories for the patients. We cannot keep depending on the pharmaceutical companies and biotechs to do this. It's going to take them too long. It's up to each one of us to do this. So what do you do in Silicon Valley? You start a company around this, right? So here's the story, right? You start with unmet need, you get the public data, NIH funding, got the story going, data analyzed, launch a company, got ARA funding, shovel-ready kind of stuff to launch, to hire the first folks, and $3.5 million Series A. Now, let me be crystal clear here in this academic audience. I'm not showing you $3.5 million to brag about my Series A. I'm showing you $3.5 million to just try to convince you this is the new way science is going to continue out of academia. We still don't know if this is going to work, but the science continues on private dollars as part of a venture-backed company, right? You know how hard it is to get a brand new $3.5 million NIH grant. You know how hard that is today. But if it's getting closer to patients, spin that out of the academic institution and get the science to continue in the company. And that's why it's not taboo to talk about this kind of thing. We got to do more of these. We got to do more of these. Not to get rich, but to address emails like this. I still get an email like this every year or so. October 8th, 2013. Hello, my name is... I need help. I'm an Italian man, 48 years old, 3 children. On June 5th, they told me I have small cell lung cancer. Neuroendocrine tumor. Today I work 75%. I have different mets. Liver, kidney, pancreas, skeletal. I know I will not live longer than a few months or over a year. My family is desperate. Please, I'd like to try the new cure that you've studied. Wants to be part of our trial. This is why we're in this business. The patients right next door and in every one of our centers continue to need help. We cannot keep waiting for others to solve this problem. It's up to us to do it. And they've given us all the data in the world to help us. It's up to us to take that on. This is an email from 2013. This guy is gone already, right? This is why I'm in this business. This is why I'm not at Google or Apple. I love those companies too, but this is why we're in medical centers, right? It's to help every one of these folks. We have a long way to go to do this. I only have a few minutes left, but let's just talk about one more story. The other story is about diagnostics. We realized that since we have all this data on diseases, let's use this to come up with diagnostics. And to be really clear, what is a diagnostic? Can I tell if someone has a disease faster maybe than others, or before older way, before just figuring out that the patient's dying of a disease? So we want to come up with more blood tests. And we realize all this data can help us with diagnostics. And how do we do this? Well, so how do we get the diagnostics? Well, we turn our crank. We got lots of stories on diagnostics. My favorite example is we had all this data on leukemia back at Stanford. And I love my leukemia collaborators. We said, look, here's a great serum protein that we computed would work on leukemia. Can you help us? Now, test our computer prediction. We go to our cancer center, right? NCI designated. Do we have a biorepository? Of course we have a biorepository. Do you have any saved samples on AML, acute myeloid leukemia, not a good leukemia to get? Of course we have samples saved. Well, they saved all the cancer cells. They didn't save any of the liquid around the cells, the serum or the plasma, right? Takes more freezers for that. And now, when I need to test a diagnostic, I go to conversantbio.com, right? Why do I need to go wait a year to go recruit my patients? Here, I can just go shopping for it. I'm in the right store. I see pictures of Accutainers. Browse by disease. What do you want to buy today? Bladder cancer, brain cancer, breast cancer. Oh, there's leukemia. Oh, yeah, here, peripheral blood plasma, leukemia. Click on that one. $55 each. How many samples do you want per patient? How many which disease subtype? And literally at the bottom again, add to shopping cart. Not the hard part. I know each one of our facilities is trying to set up biorepositories. Man, you better set up like a business, okay? Because I get this on dry ice in 48 hours, and I go forward. Last story I'm going to tell you about is the diagnostic we came up with, preeclampsia. Any of you heard of this disorder, preeclampsia? Raise your hands. Yeah, usually it's about a third of the audience. This is a third of the audience that's typically watching Downton Abbey because one of the characters dies of this disorder. Spoiler alert if you haven't seen this. And that's eclampsia. It's a preeclampsia. It's 100 years later. This is a rare kind of disease, but unfortunately not too rare, where blood pressure goes shooting high in moms who are pregnant women. And the obstetricians, I love them, but they say the delivery is the cure, so they take the baby out. The mom's better in 48 hours, and we pediatricians have to keep these babies alive for months. Kind of annoying. This disease falls right between the cracks. Forget about having a good prognostic. There's no diagnostic for this. And so we figured, let's just use our public data to do this. You turn the crank, 266 experiments already publicly available. Someone studied human samples with preterm birth, preeclampsia, prematurity. How many more do you need to get started? Now, I'm not showing you any method slides here, right? So just imagine a massive Venn diagram. What do they all see in common, right? I don't trust any one of them, but where do I see a lot of these things in common? Chase those down. And we chase it down. Here's an example of a protein called hemopexin, which is up in samples that are associated with preeclampsia compared to healthy normals. By the way, we could buy all these samples off the internet. And what do? You start a company. Now, why? This one's even more amazing. We have the unmet need. We get March of Dimes Center to get the story launched. We designed the diagnostic. We had a CTSA grant to get it launched, $50K again. Spin it out, $2 million in seed financing. Launch a multi-center clinical trial. Now, here's where it gets even more interesting. I've already exited from this company. It's already been acquired. We went from public data, which everyone thinks is valueless, to an acquired company in 24 months. Can we do more of these? You bet we're going to do more of these. Inventor happy, investors happy. And I'm giving you my secrets. There's so much unmet need, so many data sets just sitting there waiting for you, right? And so many diagnostics and therapeutics to come up with next. I'm not going to tell you about sequencing and all the rest because you've heard enough about this, so I'm going to skip those slides because I think you've seen those. I think you've seen a lot about wearables, so it's not worth talking about those. I want to just talk about a couple of things, what we're doing at UCSF next here. So we're drowning in data. I think you see that. So a couple of things in the last minute. I think it's an amazing time. California seems to be leading again. That's why I love being in that state, though. You guys are great, too, of course. So you know Obama mentioned precision medicine, obviously, in the State of the Union address back in January, but Governor Brown mentioned precision medicine a whole year earlier. So we've actually implemented state funding. The state is funding precision medicine pilots. We've launched 2 of these. It's $3 million. That's a modest amount. Even Sacramento calls that budget dust. We admit that. We're going to try to get more zeros added, but it's kind of neat that our state is funding precision medicine initiatives. I'm really pleased to be working with the governor on this. And I'm really pleased to have made this move 6 months ago to UCSF. I run this new Institute for Computational Health Sciences. We're slumming it out in that brand new building until they build our brand new building in 2019. Amazingly positioned right between, it's in Mission Bay if you've ever been there, amazingly positioned between Illumina and the brand new hospital which just opened. You couldn't buy a better spot for someone like us to be there, right in between. Some think we're going to be the wall between these two. Of course, we are going to be the conduit between these two. And in some ways, why did I move from Stanford to UCSF? Yeah, it's a great building. It's going to be great recruiting a dozen new faculty. We've got a lot of space and money for them. But it's really fun because I'm in charge of the entire University of California health system's medical records. That's UCLA, UCSF, UC, San Diego, Irvine, and Davis. It's currently 14 million patients, and we're going to build out that data repository for all of that. Going to have a lot of fun there. So last point. in Silicon Valley, I know they asked you to imagine Silicon Valley. Can I tell you how annoying it is, these kids that start these high-value companies in the garages like HP and Apple, or start high-value companies in their dorm rooms like Google, Yahoo, Facebook? I fundamentally believe the next Genentech, the next Amgen, the next Merck is going to start in a garage. When you go home to your garage, you got more than 1 million microarrays, and every mouse model you can think of probably you can order with a credit card. Yes, I know your credit limit isn't that high. That's what angel investors are for. And I don't mean making meth in your garage. That's not what I mean. I mean making the next cancer drug, the next diabetes drug, making all of those for all of us here. So what is big data in biomedicine in the end? Is it about algorithms, programmers, Hadoop, cloud computing, mobile? Yeah, it's about all of these. It's about predicting diseases before they strike, explaining rare diseases that defy experts, finding drugs for diseases that lack attention, making sure we do the right, safe, cost-effective thing for patients. And it's an amazing platform for biomedical innovation and entrepreneurship. I can sum all that up with one word: hope. I have a lot of hope for this field. I have a lot of thinking that we need more people in this field. I'm not sure about the big companies solving these problems for us. It doesn't matter. We are all going to do this in our garages one by one. Can't do any of this work without a long list of collaborators, and I have to thank the UCSF for recruiting me with endowment support. I'm currently funded on 17 NIH grants from NIH, 9 different institutes, March and JDRF, HP, HMI, a lot of stem cell and disease-specific foundations that fund us to try to find cures or treatments for their diseases. I always thank my admin and tech staff wherever I go. I'd never get a grant or paper out the door without them. I thank Zach for being my mentor, Keith and Sam for recruiting me to UCSF, and I always thank my wife, a molecular biologist, biochemist. She sees every major paper grant that comes out of my lab. She starts companies with me now and lets me go all over the country and all over the world to give talks like this. Thank you very much.